Thanks, Philip, for this wonderful session. I'll try to be quick so we all have time for a coffee break. So as explained, I'm Marcelo. I'm going to talk a bit about Molstar today. So I'm going to be a bit of an odd fish here, but let's see how that goes. I'll get Jose introductions because I think that was wonderful. PDB is part of the WWPDB. That's the group I'm in as a bioinformatician. It's also a data resource that's managed by EMBO EBI. And there is some other sister resources to PDB, which is PDB, KB, and AlphaFold. And currently, we are a team of 34 members between curators, bioinformaticians, software developers, and research fellows. If you haven't heard about Molstar before, Molstar is a modern web-based open source toolkit for visualization and analysis of large-scale molecular data. This is the URL for Molstar. And its technology is based on TypeScript and WebGL. It's released under the MIT license. And some quick stats about Molstar. It's been developed since 2017 by PDBE, RCSB, SATEC, and AlexCCZ. It builds on the progress of previous viewers, such as Lightwall and the NGL viewer. Its code is available on GitHub, and it's published via NPM. So that's how the current Molstar viewer page looks like. So you can see here there's a button for opening the viewer and some interactive examples. This is the interface of the viewer currently. So you have a 3D canvas in the center, a sequence panel on the top, some left and right controls. So on the left controls, you can, for example, load different PDB structures. And on the right control, you can representations, so both the colors and the representation types inside Molstar. One of the examples that I really like is the mesoscale viewer. So this you can load very large models. I think this model has 38 million atoms, and it's very memory efficient. And you can open this in a browser and easily navigate through all the different molecules that are inside this structure, for example. Some other members of the Molstar family, as Jose had mentioned, binary CIF is a compression format for macromolecular structure data. But we also have model server and volume server, which are tools for pre-processing and querying macromolecular structure data and volumes. And so in this example here, you can use these API endpoints for a given entry, and then you get all residues surrounding a given radius for a ligand or a residue of choice. And for volumes, we have the same thing, and also including downsampling of the data, so you can get the data in various detail levels, and this allows us to stream data very efficiently inside Molstar. Besides the Molstar, we also have PDB Molstar, which is a wrapper around it. It's been developed since 2019 by Mother first and now by Adam, whose picture is here on the bottom left. So PDB Molstar is tailored for the needs of the PDB pages and our users. It contains various predefined views and customized control panels. I'll show them in a bit. And it also has lots of helper functions for the developers that are interested in using Molstar for their pages. So on PDB KB protein pages, one thing that we like to show is the superpositions for a segment of a protein of all the structures that are available. And we have custom Molstar panels that show these superpositions here. And you can select and show them individually, and also load the output structure for this, so you are able to easily compare between these different structures. On the AlphaFold page, we also have integration of Molstar here, but with more interactivity. So we have click and hovering interactions between the PAE plot and the alpha missense pathogenicity heat map with Molstar. So it's all integrated in the same page, and we have codes to help you develop this too. So I'm going to very briefly now talk about new features that we are developing in Molstar. The first one is a headless mode, so just the ability to use Molstar as a command line tool. And also something that was added is the oriented axis feature. This allows you to minimize the occlusion of visualization by calculating a PCA of the atomic coordinates, and then you align these PCAs to the screen axis. So basically, you're laying the structure flat to the screen, and you are able to visualize more things. By combining these functionalities, we're able to create a pipeline that automatically generates images for every PDB structure. And you can also highlight individual molecules and generate images for these. Assemblage symmetry visualization. So this is a story that goes a bit way back. It started by the standardization of unique identifiers for assemblies inside the PDB archive. This is work that was done by Shri and Romana. And they also created a pipeline for automatically naming these different complexes, these different assemblies. After that, they used the Anana software to calculate assembly symmetry data for all the PDB entries and create open endpoints that anyone can use for this. And then this is a sort of visualization that was included in Molstar, so you can see different assembly information quite easily. Last, I think the most important one is MolviewSpec. The traditional way to visualize structures was you download your structure, you go to your viewer of choice, and then you do either analysis or you generate a nice figure. And what we are proposing now is a unified JSON format that would allow to be taken by any viewer, so it would increase transparency in science. And this has already been started. So this is how the commands look like in this JSON format. So you can download the structure, add representations to it, and different colors, and also different annotation labels. So you'll be able to generate images like this one. And so Adam is working now on the Python and the TypeScript library to generate his MolviewSpec files, and also to be able to load these in different viewers. So there are already lots of features that have been added. I will be happy to talk more. I have a poster on Monday about this. And I would like to thank the whole PDB team and also our funding agencies. Thank you.
Yeah, thanks very much. We talk. Any questions? So we have time for one question before we break for tea.
Hi. I think the MolviewSpec is very useful. We in the community can use the same spec for all the different viewers. I have a question. So sometimes when you apply the color or style, the order matters. So do you consider the order in the spec?
I believe the order is considered. Adam would be able to talk more about this, but I believe so, yes.
So if you use MolSpec, in general, you could generate, it's kind of a command, right? You could align all the commands together as a long URL. You can shorten the URL as a shareable link as in iCn3D, right?
Of course, I believe now we already have shareable links in Molstar. I'll have to double check, but I believe these session links are already available.
Okay, that's great. Thanks.
Thank you.